I'd like to review the use of the 2011 Onion ipmPIPE location form and then later the observation form that we'll be using to record information on crop and pest and disease development during the course of this Onion ipmPIPE project. As you can see here, this Excel sheet is laid out in this form. It has various columns that will be including information from the Sentinel plot sites. Let me go over here to the example sheet and we'll work off of this. Also, you notice down at the bottom here we have various pages that you can click on that have resource information. So for example, the FIPS numbers for the USA are shown here if you click that page. Here's some information on the reference descriptors, the host codes, and then of course here's the example sheet. Looking at this, you'll see that we have location type, location setting. The type is 1, 2, or 3. One's a sentinel plot, for example. A location setting, this would be done in a commercial field or a research plot or volunteer allium survey, could be other or weed survey as well, 1 through a 4. So 1 indicates commercial field, 2 is a research plot. Location name. This is the important column to define one time your Sentinel plot site. What we want to do is use the state name, CO for Colorado, the county name, Larimer, ST for Sentinel plot, and then 01 for the first Sentinel plot in Larimer County. Now if you see here in Weld County we have 2 sites, Co-Weld ST01 and 02. So we have 2 locations in Weld County, and then 1 site in Otero and 1 site in Mesa County. So this is a unique codifier that we can use for project communications. If we go over here, here's our FIPS numbers that I obtained for the county from the bottom of the page. You'll see the county FIPS page here. So I was able to find that number and enter it one time. Here's our contact information, the nearest city where that plot is located, geographical markers, usually roads, Road 70 and Road 37 for example. And then here's our geospatial information, that's very important, our longitude and latitude. Plot size is shown. Let me pull this page over. Okay, plot size. We also have the host code, and here you can see 1008 refers to Onion. And here's a list of all the host codes down here if you click on that page. Cultivar variety if known, and then planting date. Here's another column for additional information if you wanted to record some other agronomic notes or pest or disease notes that are relevant to that Sentinel plot. This would be a spot you could do that as well. Okay, now we've gone to the observation sheet. This is identified here as Onion ipmPIPE 2011. You see it also is an Excel sheet, and it has, again, a variety of columns that we'll review here in a little bit. Here's the location name. I just want to point this out. This is the location name and GPS coordinates that we defined in the previous Excel sheet will be carried into this area. Let's go to the example sheet here. Okay. Now this will show again the variety of information that we'll be entering. Here's our observation date. In this case I used May 5th as an example. Our state. Here's the location name. So this is our unique codifier. Here you can see the first 3 lines. We have Colorado, Larimer County, ST01. And I have 3 lines. If you come across, you'll see here's our plant stage, so on a 1 to 9 scale that's defined. Below. The organism, and here you can see I have a different organism number in each of these lines. And these are referring to 20 is Xanthomonas leaf blight, 310 refers to IYSV, and 421 refers to onion thrips that we were scouting for on May 5th at this Sentinel plot site. So I enter that information, and then simple observation information. I found, or I didn't find, the pest of interest or disease. I was scouting plants, 100 plants at each of these locations, and then I recorded a number of positive units. Let me pull this over a little bit. And here you can see the number of units. We had no plants that were showing evidence of the Xanthomonas or IYSV on this date. We did find one plant that had some thrips on it, so we entered one of 100. And here you can see the incidence of thrips per plant, rating on like a 1 to 5 severity rating. We can enter that information as well. And then we get over to here, the confirmation method. You'll see we have different numbers. Again, those are defined down here in this page. And you can see we have a unique number for the various methods that we can use to evaluate for presence of that organism or insect. Using visual rating of 3, for example, or if we use some lab technique, ELISA or PCR, we would enter that information here for fungi, bacteria, and nematodes. Viruses, we have another list here, 11 through 16, in which we can also use ELISA, but we want to just note that this was ELISA for presence of a virus, such as IYSV. We can also note PCR and other types of activities. For insects, we just have 2 diagnostic approaches at this point. Our field identification visually or a lab identification, number 18. Let's go back to the form here. And then finally, we also want to note if that sample you collect in the field was also submitted to a diagnostic lab for confirmation, and enter no or yes. If it was entered, and if they assigned, which they should, a unique number to that sample, please record that as well in this column, so that we have a cross-reference between their diagnostic lab approach and our onion ipmPIPE information. So that concludes the information on the observation sheets that we'll be using this year. One other point, each time that I go into a field, for example, and make another observation, so for example 2 weeks after May 5th, I will just copy and paste this information down below. So for example, I would copy this, come down here, paste it. I would also include the other information on these lines, and then I would just enter, I would change the date and enter the any changes in organisms I was scouting for, plus the new information. And I retain the original scouting date with my next scouting date and continue this on this form. So this way, at the end of the season, I may have 5, 10, 20 groups of data like this complete from the beginning of the season to the end of the season that allow me to record and have this archived record of the information. And this is what you will then upload to Planalytics to add to the website, and from which they'll also graphically portray our pest information in the upcoming weeks. Thank you for your attention.